These videos are offered on a pay-what-you-like basis. You can pay for the use of the videos at my website. There's a link to my website in the annotation. The address is www.freelance-teacher.com/videos.htm. Or you can just use the link in the annotation. Thank you. So to begin with, what are the three basic stages of signal transduction? What are the three basic stages of signal transduction?
Reception, transduction, and response.
So now we're going to be going through an example of that process. So when a plant is in the dark, it's going to grow differently than when it's in the light. So the plant needs to know when it's in the dark and when it's in the light. Let's see, one moment. So for example, if the plant's in the dark, it might be still, say, underground in the soil. So for example, while the plant is in the dark, it's not going to be growing and expanding leaves, right? There's no point in expanding your leaves while you're in the dark. Obviously, if you're in the soil, you don't need leaves. And even if you're above ground, the leaves won't do you much good if you're in the dark because they're not going to be able to absorb any sunlight anyway.
Yes.
So for example, while you're in the dark, we don't want leaves to grow. So what should the plant do then when it senses light? Well, when the plant senses light, it should start growing and expanding its leaves. So that would be, and those would be green. So that would be one example of the light response, which is also called greening. The plant is not going to be producing a lot of green leaves while it's underground in the dark. But when it receives light, it is going to start producing. Those green leaves. And there's other responses. Obviously, there's the plant is going to grow differently when it thinks it's in the soil than when it thinks it's above ground. So let's say that the plant starts in the soil, and then suddenly there is a light signal. Well, obviously, the plant is going to want to change its behavior now that it's getting that light signal, so we can go through the process for that. So we have a light signal. So here's some light, and the light is going to have to be received. Remember, the first step is reception here, and I guess you're going to have this memorized, right?
Yes.
Okay. Do you happen to remember what is the first molecule in this process that receives the light signal?
The phytochrome.
Okay, very good. So.
And specifically, what inside the phytochrome would be the kinase?
The who?
The kinase inside the phytochrome is what?
Oh, the kinase. So it's actually not the kinase that actually receives the signal. The kinase is not actually going to receive the signal. The part of the phytochrome that's going to receive the signal is what's called the chromophore.
Oh yeah.
It's a pigment.
Myself.
So the phytochrome has a pigment. A pigment is something that can absorb light. Do you know what a kinase is? Or what do kinases do?
It's what triggers the transduction.
Okay, actually, this will be important. A kinase, first of all, is an enzyme.
Okay. Yes.
A kinase is an enzyme. Maybe we should take a detour. When a word ends in "ase," what type of molecules end in "ase"?
Enzymes?
Yeah, ACE means enzyme. And remember, what are enzymes? Enzymes are a type of protein. They're a type of molecular machinery that do things. Enzymes help reactions to occur. So a kinase is an enzyme that adds a phosphate group to another molecule. That turns out to be a very important process. When you add a phosphate group to another molecule, that often changes the other molecule's behavior. Okay. So when something ends in ACE, what types of molecules end in ACE?
An enzyme?
That's right. What's a kinase?
Kinase is the molecule that it's what triggers an action to happen.
Let's be more specific. A kinase adds a phosphate group to another molecule. Okay. A kinase is an enzyme that adds a phosphate group to another molecule. Kinase. It's important to know that specific definition because those play an important role. Okay, so what type of molecule is a kinase?
Enzyme?
That's right. It's an enzyme that does what?
It's the enzyme that adds a phosphate group to another molecule.
Okay, that's right. Now, it's true that phytochromes have a kinase function, but that's actually not going to play a role at the beginning of this process here. So that was actually a little bit of a detour. Instead, the light is going to hit a pigment on the phytochrome, and the light is simply going to be absorbed. And what happens when we do that is we get a activated phytochrome. So I'm going to use an asterisk to show when something is activated. So the light hitting the phytochrome activates the phytochrome. It causes the phytochrome to change its shape in a way that it can carry out the next step. So the next step of this process, so which part of the process have we done so far? Reception, transduction, or response?
Reception?
This is reception, yeah. So this step here, when the light hits the phytochrome, this is reception. This cell is receiving the signal that there's light outside and it should start its greening responses. It's not in the dark anymore. Okay, now we're going to go through the signal transduction. So the key thing to realize here is, do you know what a Rube Goldberg machine is?
No, I do not.
So a Rube Goldberg machine is something where you set up A to do B, and then B does C, and then C does E, and then E does F, and then F does G. Oh. It's like a long domino chain of one thing causing another thing, causing another thing, causing another thing. So for example, you might say, drop a ball onto a lever. That raises the lever up. That tips over a glass of water. The glass of water fills up another glass of water, which raises another lever, which pushes over a domino, which pushes down a string of dominoes, which hits a light switch, which turns on a light, which is detected by a camera, which sends an electrical signal. So the point is, a Rube Goldberg machine is when you have a whole chain of different machines hitting each other and causing a huge sequence of effects. Anyway, the point is, that's what almost all of biology is about, Rube Goldberg machines. So almost all of the processes in the cell involve one thing causing something else to happen, causing something else to happen, causing something else to happen. So it's kind of a pain when you're trying to prepare for exams to learn the whole thing. But anyway, that's what we're going to look at now. We're going to see a little kind of a complicated Rube Goldberg machine. So the light has changed the shape of the phytochrome. Now the next thing is that the phytochrome is going to bump into what's called a G protein. So here we have a G protein, and the G protein is connected to a molecule called GDP.
Okay.
And this is a inactive G protein.
Mm-hmm.
Why is it called a G protein? The G refers to the GDP. It's called a G protein because it's bound to GDP. So the phytochrome now is going to attach
okay.
to the G protein.
Got it.
The activated phytochrome attaches to the G protein. Okay. And that allows the G protein to exchange GDP for GTP.
Okay.
What if they just go in demonstration on this?
Set fire.
Hold on. I'm trying to look up this example in the textbook and I can't find it. Okay.
This doesn't make sense.
Okay. So are you with me so far? The phytochrome allowed the G protein to exchange GDP for a different molecule that's called GTP. So notice the new GTP came in and the GDP has floated away. And notice now I put an asterisk on the G protein. Remember that asterisks means that this has been now activated. The G protein is now activated to carry out the next step in the chain. Okay, is this making sense so far?
Yes. The only thing so the GTP is how does it know? It just knows when the phytochrome is just activated that it needs to attach to the G protein so the GTP leaves.
Well, yeah, so that's a good question. Of course, nobody really knows anything because these are not people. They don't have conscious. Oftentimes, it helps to understand things if you pretend that they're like little people. But here's what's really happening. What's really happening is that we just have a bunch of molecules bouncing around in the water. We just have a bunch of molecules bouncing around in the water. So G proteins are constantly bouncing into phytochromes. But normally, the phytochrome is normally not the right shape to attach to a G protein. Normally the G protein bounces into the phytochrome and then just bounces off with nothing happening. However, when light hits a phytochrome, that activates the phytochrome. It changes its shape so that now when the phytochrome accidentally bumps into a G protein, now they can stick together and fit together. So it's not like the G protein saw that the phytochrome was activated. It's just randomly bouncing around. But now that the phytochrome is activated, the next time a G protein bounces into the phytochrome, they're going to stick and fit into each other. Also, GTP molecules are constantly floating around and bumping into G proteins. But normally, the G protein is not the right shape. Normally, the G protein is not the right shape to exchange GDP for GTP. Normally, even though they bounce into each other, they just bounce off. They're not the right shape for anything to happen. But when a phytochrome is attached to a G protein, that changes the shape of the G protein. So now the G protein now is the right shape. So the next time it accidentally bumps into a GTP, now it's the right shape for the GDP to float away and the GTP to stay stuck to the G protein. So that's what's really happening here. What's happening here is that things are just randomly floating around and bumping into each other. Normally, nothing happens when they bump into each other, but when something has become activated, then it's the right shape to fit with something to fit with the other molecules when it bumps into them, and for an interesting reaction to happen. Did that answer your question? Does that help?
Yeah, yes, I understand. I wasn't sure. I realize now that they're just attaching. They're not necessarily being activated at that point, but when they do hit the G protein, it will be activated.
Okay, I hope I followed you there. So maybe as we continue to go, the point of this will become clearer. And obviously, after we go through it once, we'll have to review it. So the point here is now the G protein has lost its GDP and replaced it with GTP. And notice that now I put a star here to show that the G protein has now become activated. So the G protein now is going to continue floating along, and it's going to bump into the next participant in this Rube Goldberg mechanism. Now the G protein is going to bump into what's called guanyl cyclase.
Okay.
So the activated G protein and the guanyl cyclase are going to bump into each other.
So I want to see what happens. This is like for.
No,
So like, that's what happened. Usually we're just looking at the concentration of the S2O. So I would write like.
Now this guanyl cyclase here is inactive. This is an inactive guanyl cyclase, but when it bumps into the G protein, that changes the guanyl cyclase's shape so that now it's activated. It can carry out its function. So I'm going to put a star here to show that now the guanyl cyclase has become activated. Bumping into this activated G protein changes its shape so that now it can bump into something else and cause the next step in the reaction.
Okay.
So if you look at.
Okay. Is this clear so far?
So far, yeah.
Okay, so now we're ready for the next step. So what is this activated guanyl cyclase going to do? Well, it's going to take another molecule of GTP and it's going to turn it into what's called cyclic GMP. So notice here what's happening is the guanyl cyclase turns GTP into cyclic GMP.
Okay. Is that just the secondary messenger?
Very good. This is what's called the second messenger.
Okay.
Because it's carrying the signal.
So now your equation is broken.
Okay. So then the cyclic GMP just goes floating off.
K times S2O into.
Okay. So tell me if that's making sense so far.
That is making sense.
All right. So then the next step is that the cyclic GMP is going to bump into a protein kinase. I'll call this protein kinase A. That's not an official name. That's just a name I made up for it.
And that's just
solve for K, you have to rate, and do it for.
And they can attach to each other.
They give you these values. At this point, when you plug in all you do solve for K, you just do it for all.
so you plug in.
You're just using this.
Actually, one thing I should say here is the guanyl cyclase is probably going to make many of these cyclic GMPs.
Okay.
It can take a GTP and make it into cyclic GMP. Then it can take another GTP and make it into another cyclic GMP. Then it can take another GTP and make it into another cyclic GMP. So it's amplifying the signal. And then each of those cyclic GMPs, they could attach. They could attach to a protein kinase. And now I'm going to put a star on the protein kinase. Do you remember what are we using the star for?
When it's activated.
So now the protein kinase is activated. So the protein kinase is going to go floating off, and it's going to bump into another protein kinase, which we could call B. That's not the official name, it's just another name. Now, do you remember way back a long time ago, we were talking about what kinases are? Do you remember what type of molecule is a kinase?
Enzyme?
They're an enzyme. That's right. Enzymes do things. What do kinases do? What does a kinase do?
It adds a phosphate group to the other molecule.
That's right. So what is now that it's activated, what is protein kinase A going to do to protein kinase B?
It's going to add the phosphate group?
That's right. Okay, very good. In fact, it could do that many times. a single protein kinase A might phosphate 10 or 100 or 1,000 or 1,000,000. I don't know how many times, but it could phosphate many, many protein Bs. And now that this is phosphated, I'm going to put a star on it. What does the star mean?
It's activated.
It's activated now. So what is this protein? What is this? Kinase B going to do? Well, that might float away until it bumps into another kinase. I'll just call that kinase C. That's not the official name, but it might bump into kinase C. And what could kinase B do to kinase C?
Adds a phosphate group to another molecule.
Well, what would kinase B do to kinase C? So my point is that it would add the phosphate group to kinase C.
Yeah.
It bumps into it. Okay. I just wanted to make sure that was clear.
Oh, okay.
So now this will have a phosphate group. Now I'm going to put a star on kinase C because now it's activated. So what could kinase C do now?
It's gonna add a phosphate group to kinase D.
Yeah, and then what could it could do that many times. It could phosphorylate 10 or 1,000,000 kinase Ds. And then what could one of those kinase Ds do?
It'll just continue.
So let's take it one more step to make sure we understand. So what could kinase D do?
It could add the phosphate. Well, it becomes activated and then adds a phosphate group to kinase E.
Okay, good. Well, obviously, we don't have to keep carrying out this anymore at this point. So what's the name of this part of the process? This is what's called a protein kinase cascade. This part of the process is called the protein kinase cascade. You see how that's a logical name because it's a cascade of effects. A could phosphorylate 1,000 Bs, and then each B could phosphorylate 1,000 Cs, and then each C could phosphorylate 1,000 Ds, so we're amplifying the signal at this point. The signal is getting more and more amplified. Okay, all right, so this is obviously not going to go on forever, so I'll just say ... Eventually, we're going to get to the end of the chain. So we could call that maybe kinase Z, just to give it a name. I'm not saying there have to be literally 26 in the sequence, but let's just call this kinase Z, and it's got a phosphate group now attached to it. So it's got a star. Now, where has all of this been happening? Where have all of these processes been happening? I should have mentioned this before. All of these processes are happening in the cell's cytoplasm. Do you remember what the cytoplasm is?
Yes. It's the spatial. It's like the spatial area inside the cell where there's no organelles.
That's right. So is it inside or outside the nucleus?
Outside the nucleus.
Yeah. So all of these steps that we've been talking about so far are occurring in the cytoplasm. All of these steps are occurring
Is this only one of the transduction pathways?
That's right. Yes. So this is very complicated. There's actually 2 different transduction pathways. That's right. So we're still trying to finish up one transduction pathway, and then after we do that, we'll have a whole different one to take a look at. But yeah, so this is one of the transduction pathways. So we're nearing the finish line for that. So then this kinase Z here could enter into the nucleus. So now we're going to enter the nucleus of the cell. What's the most important thing that lives in the nucleus of the cell?
The chromosomes?
Yeah, the chromosomes, which are made out of DNA. So the nucleus is where the DNA is. Okay. Now, the DNA consists of genes, right? And what are the genes? The genes are information that tell the plant what proteins to make. The DNA has genes, which has information that tells the plant what proteins to make. That's something you probably covered in the first semester of OChem when you studied, of biology when you studied genetics. So the DNA has lots and lots and lots and lots and lots of genes. So inside the nucleus, we have a DNA molecule. Which has lots and lots of genes. So only some, do you remember the process by which DNA, that DNA produces the proteins? Do you remember from the first semester there was transcription and then translation?
Mm-hmm.
Okay, so a portion, one of the genes will be transcribed and then translated into a protein. How does the plant know which genes to transcribe and which genes not to transcribe? Well, there are little molecules floating around which are called transcription factors. So when a transcription factor binds to a portion of the DNA, that tells the cell to transcribe that portion of the DNA. And when a different transcription factor binds to a different portion of the DNA, that tells the cell to transcribe that portion of the DNA. So what's going to happen here is that the kinase is going to enter into the nucleus. And the kinase is going to do something to this transcription factor. What would be the logical thing for a kinase to do to a transcription factor? What do kinases do?
Oh, it adds a phosphate group?
Yeah, so who is this kinase going to add a phosphate to?
To the transcription factor?
That's right. Notice how this is finally we got to the end of the kinase cascade. In the middle of the cascade, one kinase was phosphorylating another kinase, which was phosphorylating another kinase, which was phosphorylating another kinase. When we're at the end, the kinase enters into the nucleus, and now it doesn't phosphorylate another kinase, it phosphorylates the transcription factor. This was the end product here. Okay, and this is the portion now here which is considered the response. This is where the cell is actually going to respond to the signal. Remember the three stages were reception, transduction, and response. So now we're at the end. We're going to have the response. This phosphate group is going to activate the transcription factor. So I'll put a star here to show that this is now activated. And what is the transcription factor now going to do? The transcription factor now is going to bind to a section of DNA. It's able to do that because it's been activated by the phosphate group. What happens when the transcription factor binds to that portion of the DNA? Well, then this portion of DNA is now transcribed. Why is a transcription factor called a transcription factor? It's called a transcription factor because it causes a piece of DNA to be transcribed. If you remember, when the DNA is transcribed, it turns into mRNA. And then that mRNA exits the nucleus and enters the cytoplasm. And remember what happens when it reaches the cytoplasm. That's when you then have the mRNA is translated by the ribosomes. You studied that process in the first semester. mRNA is translated, and that produces the greening proteins. Those are the proteins that are going to cause the response to the light. So for example, these are proteins that will allow the plant now to start growing green leaves now that it's in the light. And that's the end. That's the end of the story. So it really is this huge, long Rube Goldberg machine that is quite, well, it's quite a long story here. Okay, so to remember this whole process, remember we're trying to go over the process by which plants respond to light. The process by which the plant responds to light. So let's just make sure that we understand all of these ideas. So right now, obviously I don't expect you to have this memorized yet. The first step is to understand. After you understand, then you can memorize. So right now, I don't want you to rely on your memory. Let's just make sure we understand what's happening here. So what part of the molecule receives the signal from the light?
The phytochrome.
That's right. And you can just go ahead and look at your notes or look at my screen. I just want to make sure you're understanding what's in the screen. Okay, good. So the light is going to hit the phytochrome. Good. Then what?
Once the light hits the phytochrome is activated.
Good.
And then the inactive G protein is going to attach to the activated phytochrome.
Good.
And then the GDP will attach to that G protein.
Say that last thing one more time.
And then the GDP is going to attach to the G protein.
Now, actually, what's happening is the GDP is leaving the G protein. And GTP is attaching.
Oh, the GDP is already it's already attached to the G protein.
So yeah, so let's clarify that. The inactive G protein has GDP. An active G protein has GTP. So the way to activate the G protein is to kick off the GDP and replace it with GTP. Okay, so I didn't really clarify that before. All right, so let's try that again. So the phytochrome has been activated. The phytochrome is going to attach to the G protein, an inactivated G protein. And then what happens now that the G protein is connected to the phytochrome?
The GTP is going to kick off the GDP.
Yeah, the GTP will replace the GDP. Good.
And then the G protein will be activated.
That's key. Having the GTP now means that it's activated. So the phytochrome can now float away. By the way, where are all these steps happening? Which part of the cell?
Cytoplasm.
Yeah, this is happening while things float around in the cytoplasm. Great. Okay, so now we have an activated G protein. So what's going to be the next step?
The G protein is going to attach to the guanyl cyclase.
That's right. It's going to attach to the guanyl cyclase. Good.
And that will activate it. And
Who's
then,
it?
I'm sorry?
You said that would activate it. Who's it? Who's
Yes.
going to be activated?
The guanylin. I can't pronounce that cyclase.
Yeah, the guanyl cyclase. So by the way, one thing we should work on here is when you're reviewing this material, the one word you never want to use is it or them. Because what the grader is going to be looking for is using the correct terms and the correct names. So as we go through this, we want to at each step always make sure we're using a specific name, and we don't want to say it or them. We want to make sure we know exactly who we're talking about. But what you said was correct. The G protein will attach to the guanyl cyclase, and that activates. That activates the guanyl cyclase. Good. So then what?
The GTP of the guanyl cyclase will activate the secondary messenger, cGMP.
Okay, that actually isn't quite right. I don't think I quite explained what was happening here. Right. So what happens here is that the guanyl cyclase will transform GTP into cyclic GMP.
Okay.
So I suppose what happens is that the GTP will be briefly attached to the guanyl cyclase, and while it's attached to the guanyl cyclase, it will turn into cyclic GMP, and then it'll be released as cyclic. So I got a little lazy and I skipped some steps. So what I was trying to show by this arrow is that the GDP will attach to the guanyl cyclase, it'll turn into cyclic GMP, and then it'll be released as cyclic GMP. And that will happen many times. Then another GTP will attach to the guanyl cyclase and turn into cyclic GMP. And then another GTP will attach to the guanyl cyclase and be turned into cyclic GMP. So that can happen many times. Okay, so when you're ready, let's review that step. What does the guanyl cyclase do? So the point we were at was we have an activated guanyl cyclase. What does the guanyl cyclase do?
The guanyl cyclase will become the cGMP.
Let's actually review that again. Maybe, I guess this is not a very good picture. The guanyl cyclase is not becoming the cGMP. The guanyl cyclase is turning GTP into cyclic GMP.
Okay.
Maybe let me write this out so that it's clear. Oops. So these two things will come together. So now we'll have
You're gonna take sour. I'm gonna go walk back and take a shower real quick. And then I'm gonna come right from here. I was gonna say, we'll sit more left. Yeah, I'm not gonna have a shower right now. I need challenges like yesterday at closet. There's no shower, so we should
You shower in the middle of the day. I either shower at night or
Because
before.
of that.
So it was not doing it. It's small shit costs. It's not really shower. It's just small shit all day.
Yeah, but,
Shower. The table is disgusting.
yeah.
Okay, so I'm trying to write out more clearly what's happening here. The guanyl cyclase attaches to the GTP, and it turns the GT. Well, I still didn't do it clearly enough. So it turns the GTP into cyclic GMP, and then it releases the cyclic GMP.
Yeah.
Okay, so I hope that and the cyclic GMP could be considered the activated form here. All right, I hope that's a little bit clearer. So what we have here is a activated guanyl cyclase. What does the guanyl cyclase do once it's activated?
Okay, the guanyl cyclase will become it will allow the GTP to become the cGMP.
Good. Okay, that's it. And then the guanyl cyclase will turn another GTP into a cyclic GMP. And then the guanyl cyclase, so this whole process repeats many times. So you'll produce many of these cyclic GMPs. By the way, do you know what does this little C stand for here? Remember, this is what we call the cyclic GMP.
Cyclic, yes.
So what does the little C stand for?
Cyclic GMP.
So why do you think that this substance is called a cyclase? What does it do? It makes GTP cyclic.
Okay.
That might just help you to remember this a little bit. This is not cyclic, but the GTP is turned into a cyclic molecule by the cyclase. And by the way, this G stands for guanine. So guanyl cyclase is something that takes guanine and turns it into a cycle. Guanyl cyclase takes guanine and turns it into a cycle. Okay, so then what's the cyclic GMP going to do?
The cyclic GMP is going to it's going to attach the protein kinase A and activate it.
Good. And of course, A is just a name I made up just to help go along with the series, but that's good. And then what could protein kinase A do?
It'll add the phosphate to protein kinase B.
That's right. And then what could protein kinase B do?
It'll add a phosphate to protein kinase C.
So what's the name of this whole process that's occurring now?
Phosphorylation cascade.
Yeah, the protein kinase cascade. Good. Where is this occurring? Well, all right, this is probably still in the cytoplasm.
Mm-hmm.
What happens when the the kinase cascade finishes?
The kinase Z, I guess you would call it, would enter inside the nucleus.
Good. And what is kinase Z going to do in the nucleus?
It'll activate the transcription factor.
Good.
It'll attach a phosphate to the transcription factor.
That's better, because that's what kinases do, right? Kinases attach phosphate groups, so it'll attach a phosphate group to a transcription factor. Okay, excellent. Then what will the transcription factor do?
It will attach. It will begin the transcription process, transcription of the DNA.
So yeah, so it attaches to a section of DNA. It attaches to one particular gene on the DNA. And then that portion of DNA will be transcribed. And then what?
And then the mRNA. Okay, once it's transcribed, it becomes mRNA. And mRNA, it leaves the nucleus, goes into the cytoplasm, and then mRNA becomes translated, and then that creates the greening proteins
that are going to cause the response to the light, like producing the green, like producing the green leaves. Okay. So it's a pretty complicated Rube Goldberg type of situation. So this whole process is initiated by light. What does the light do?
The light activates the phytochrome.
That's right. So here's the phytochrome. And then what does the phytochrome do?
The phytochrome activates the G protein, and then the GTP kicks off the GTP.
Well, okay, yeah.
Sorry.
Okay, I'm sorry. I think you were doing pretty good. So let's say again, what does the phytochrome do?
The phytochrome attaches to the inactive G protein.
Good.
And then GTP will kick off GDP and then activate the phytochrome. Is that correct?
Say that one last part. The GTP will kick off the GDP. Good. And that activates who?
The G protein?
Yeah, not the phytochrome, but the G protein. Good. So now that we have an activated G protein. Good.
And then the activated G protein will attach to the guanylyl cyclase.
Good.
And then that guanylyl cyclase will, oh, no, not the guanylyl cyclase, the GTP will become cGMP.
Who turns the GTP into cyclic GMP?
Is it the phosphate? No. Who turns the guanylyl cyclase?
That's right. Yes. So it looks like you're having a little difficulty interpreting this type of arrow.
Yeah.
So what does this arrow mean? This type of arrow means GTP briefly attaches to guanylyl cyclase. While it's attached, it turns into cyclic GMP, and then it floats away. So what that little arrow really means is what we were talking about here. The GTP attaches to the guanylyl cyclase. While it's attached to the guanylyl cyclase, the guanylyl cyclase turns it into cyclic GMP. Then the guanylyl cyclase releases it, and it floats away as cyclic GMP. So let's try again. What does the guanylyl cyclase do?
The guanylyl cyclase is what TP into the cGMP.
I'm sorry, there was some static. Can you repeat that?
The guanylyl cyclase will turn the GTP into the cGMP.
Okay, good. What is guanylyl cyclase? What type of molecule is guanylyl cyclase?
It's an enzyme.
That's right. How can you tell?
It has the ACE
That's
at
right.
the end of it.
Remember, enzymes do things. So what does guanylyl cyclase do?
Guanylyl cyclase will add the phosphate group to another molecule.
I think you're getting a little confused. You already gave me the right answer a moment ago. What does guanylyl cyclase do?
It'll turn the GTP into the cGMP.
Good. What does the C stand for in cGMP?
Cyclic.
So why is this called a cyclase? Because it makes GTP into a cycle. That can help you to remember the sequence here. And what does the G stand for in GMP? It stands for guanine. So what does guanyl cyclase do? It takes a guanine and makes it cyclic. Hopefully that can help you to remember the sequence here a little bit. Okay, so now we've produced cyclic GMP. What happens next now that we've produced the cyclic GMP?
When the cyclic GMP is created, that activates the protein kinase cascade, which means that a phosphate will be added to kinase A.
Good. Actually, the first at the beginning, the cyclic GMP attaches to protein kinase A. And then what does protein kinase A do?
It becomes activated,
Good.
and that sends over a phosphate group to protein kinase B.
Good.
And that protein kinase B, when activated, will send over another phosphate group to protein kinase C.
Good. Okay. Good. Then what? But now what happens when this protein kinase cascade is over?
Kinase Z would enter inside the nucleus.
Good.
And then it'll become a transcription factor, and that transcription factor.
This actually does not mean it's becoming a transcription factor.
Yes, the protein kinase will add a phosphate to the transcription factor.
Right. So notice that this started inactive, and now it's got the little P attached to it. Okay? Then what?
And then it'll attach to a piece of DNA.
Yes, this is a strand of DNA. Good.
And then that will start this transcription process, and then it'll create mRNA, which leaves the nucleus and enters the cytoplasm, and then that's when the greening process. Well, that's when the plant, the greening response.
So the mRNA will get translated.
Yes.
And what happens when you translate mRNA? That turns into proteins. That's something from the first semester. When mRNA is translated, that produces the proteins that will cause the greening response. Okay, so you can see this is quite complicated. So what I tried to do was kind of unpack some of these steps to try to explain what was going on here a little bit more clearly. Here's the step that especially gave us trouble. What was happening in this step?
In that step, guanyl cyclase is turning GTP into cGMP.
Excellent. So that's the one step that was especially giving us trouble here. So now we have a better understanding of that. And what happens in this step?
That is when the kinase Z moves into the nucleus, and then it adds a phosphate to the transcription factor.
Okay, very good. Okay, so those were the two steps that gave us trouble. Otherwise, we were getting this right. So basically, obviously, there's a lot of memorization here. All I was trying to do is help you to understand what's going on. All right, so that's something you can work on your own. Notice this is not something you can just remember automatically. It takes a lot of work. So basically, what you should be doing is you should be kind of doing what I was trying to do here. Start at the very beginning and try to just diagram the whole thing out like you would have to do if it was on the test. Try to obviously, you wouldn't write a great big monster diagram like this for an essay question. But the point is, try to think about how you would explain what's happening in each step. You can make little diagrams to try to summarize that, but you need to be able to explain what's happening at each step. So I guess actually what I should say is what you should be doing is memorize this picture, and you also have to memorize how will you explain what's happening at each step. So if this was an essay problem where they ask you to explain signal transduction for the greening response, a good way to do this would be draw this picture and then describe what's happening at each of these steps. So it's important to go back and really carefully review what we just talked about so that's clear to you. There's actually a lot of these different processes in biology, so it takes some work to memorize those, but hopefully we clarified a little what that means. Does this make any more sense now?
Oh, it makes a lot more sense now. I understand majority of it now. Like before I didn't really get what was happening when the guanyl cyclase was turning GTP into cGMP, but now it's more clear.
Okay, I hope so. Okay, so then you can continue working on that and memorizing that on your own. Now, the big thing we talked about last time, remember, was the greening response. We spent a long time going over that very complicated signal transduction process for the greening response because you said the instructor warned you there would be a question about that on the test, right? So did you get a chance to try to memorize that process? Remember that one thing we really spent a long time on last time was the signal transduction pathway for the greening response.
Yes.
Because your instructor warned you that there would be a question about that on the test. So,
Yeah.
and the test is tomorrow, right?
Mhm.
All right, so we want to make sure you'll be prepared for that question. Did you get it? So that was a really involved process. Did you get a chance to try to memorize that process?
Yes.
Okay. All right. Well, why don't we then proceed to try to review that? So, if there was a question that said, "Describe the signal transduction process for the greening response," just describe to me what your essay answer would look like.
Do you mind if I draw it out?
That sounds excellent. Great. So let's try turning on your screen sharing.
But I see we did a lot of like. Yes. And the fact that I'm just like not really million times better. Yeah. It was like I look forward to that more than a lot of other things. Just like different setting, which I feel like he's trying to change the setting of the classroom by moving the chairs.
Wait,
I don't know if that's like one
I think I may have need to erase this.
Sure. That's fine.
I just need more.
Yeah, that's fine. Great. That's one good thing about working on this program. We can always scroll to give ourselves some more room. Good.
I apologize if that's a little small.
That's fine. I know what you're talking. I know. I can see what you're doing. That's good. Good.
was like so nice and pretty. So
He's like, he felt like he hasn't had the classes
Just trying to remember the process. Hold on.
Okay, good. You're doing good so far.
Yeah.
It's a long process.
Well, I mean, yeah. It was
Oh, yeah.
It was
Wow.
basically.
But this is an important step.
Yeah. I think this we talked about this already.
She was kind of a few meetings with her beforehand about what we want to get out of it. Like, we were trying to get someone a resume, which wasn't necessarily my goal. It was just kind of like see what's out there. I'm still a sophomore, but we don't have to like huge steps. Yeah, I didn't really feel the need to like advocate myself and stuff like taking it on. So I list of like all the speakers, all the vendors who's going to be there and stuff, and it was more business oriented kind of, and I'm in like healthcare field, so it was kind of it was kind of like a different perspective. But there was an orthopedic surgeon who came. She did like a 4-hour. She talked about in terms of like sleep and nutrition. It was like interesting. It wasn't necessarily stuff I did in class, but like it was cool to see it like actually practice and real. I got to talk to her after, which was really awkward. She was there's only like under 50 female orthopedic surgeons in the whole world. Yeah. It's like it's kind of like engineering. It's a field where like yeah. And I'm shadowing an orthopedic surgeon this summer, which is like kind of cool. So we all tie together. But instead of John said that it's like even just talking to other people outside of the school, seeing us. Yeah, that's basically that means the system. She's there's like things like that. She's like, well, they were talking about possibly getting a picture. Yeah. Just like little things like that, where setting out your plan. It was a long day. The tickets are like $1,000 each school. There's like 8,000 women there or something like that. It's kind of like a feminist conference, I'm not gonna lie. Because it talks about like taking positions in corporations sort of thing, but it's cool. Interesting. We actually have to, every non-conference, we have to do like a video. We have to record like our reactions to the conference kind of thing, because it was that cut for funding, so so it's in part 362 class. We were talking about it was like this year, so we could do the same topic. It's funded. This past year, our video was kind of like to show money towards it, but it was actually worth it. So I guess you gotta do, which is cool. But like
Should I just like just continue on to the next step instead of just continue making these?
Yeah, that's fine.
Okay.
it's kind of what you make out of it. Like you could, there was a meeting ball where they had speakers going throughout the day, like would speak in the morning, but outside of that hall, there was the vendors and like it was separate forums, so like you kind of had to go out and like point down who you wanted to see and where you wanted to go. Yeah. You just have to like get there to like walk up to somebody like ask questions. This is what yeah, there was like life coaching people there, which was kind of cool. Like you could set up an appointment and do like it didn't really help because they were just like you don't really have much to tell you. There were a bunch of adults there who really wanted like business advice or like relationship advice and began stuff. Just like it was all sorts of like cool. Actually, there's kind of more stuff. Like in the video, like of the reading article, I was saying I wish there was more stuff that was being students, but for like medical field, because a lot of times it's just like image of doctors talking about this life and stuff like different skill levels, kind of like different backgrounds. Like a gap I would be able to go to. What's the I think the Pennsylvania Women's Conference. It was in Philly last year, I'm pretty sure it's
Okay. I believe I got everything. Hold on. Hello?
I'm here. Yeah, I'm here. Okay, done?
One second.
Excellent. Very good.
Okay. Okay, attaches. Yeah, okay. I'm finished.
Okay, very good. That's excellent. That shows very good progress. I'm going to go back to my screen for a moment.
All right.
Okay, so remember that we're starting this process while the plant or the seedling is in the dark. Maybe it's in the soil. The specific example is a potato that's underground in the soil.
Okay.
So how is the potato going to be growing while it's underground in the soil? Well, for one thing, there's going to be rapid stem growth. I think it's going to shoot out a bunch of different stems. Rapid stem growth. What's the purpose of that? What does the potato want to do while it's underground in the soil? It needs to reach the air. It needs to get out. It needs to reach out of the soil. So it's going to send out a bunch of thin stems to try to find where the soil ends, basically. So the potato is underground. It's going to be sending out a bunch of rapid stem growth to try to get those stems out of the soil. So it's going to have no leaves or very minor leaves because there's no point having leaves while you're underneath the ground.
Yeah.
It's also not going to be growing its roots very much, because the purpose of roots is to bring in nutrients, but it doesn't need very many nutrients at this point. While it's underground, it doesn't need very much nutrients, so there's not going to be much root growth. Instead, and it's not going to be producing chlorophyll, because remember, the purpose of chlorophyll is to absorb sunlight. Well, while it's under the ground, it doesn't need that. So this is how the plant is the plant's adaptations to being underneath the ground in the soil. Now, what's going to happen when the stems penetrate the soil and move into the air? Well, when the stems penetrate the soil and move into the air, the cells are going to start receiving light. And that light is going to start hitting the phytochromes, and that's going to begin that greening response we were just talking about. So let's go back to your screen now. Back to the beginning of the process. Scroll to where you started your process.
Oh, should I have displayed where the transduction was taking place in response?
That would be good. Yeah. So which portion we can say? Yeah. Which portion is the response section?
Oh, the response.
Oh, I'm sorry. Excuse me. I misspoke. I meant to ask which portion is the reception.
Oh. So basically, the reception is. Nope. Basically, when the reception is the light hitting the phytochrome?
That's right. So you might want to put the bracket around the phytochrome too. The phytochrome is what's receiving the light. So the reception stage is when the phytochrome receives the light. Okay, good. And then when does the response begin?
Okay. That happens. After the cascade. It's when the last, whatever letter of the kinase or whatever, how many have been made, that's when that kinase enters the nucleus, which is here.
And activates the transcription factors. Good. So that might be a good thing to include in your answer, because that's one of the main learning objectives here. So that's when, according to the textbook. Now, I think you wrote down an incorrect word there. Reception was at the beginning.
Oh, whoops, sorry.
It's easy to confuse those because they both start with res.
I mean, I knew it was response, but for some reason I just wrote out reception. Wow.
Anyway, it's easy to make that careless mistake, so you want to watch out for that on the test. Always good to double check yourself. Okay, good. And everything in the middle is transduction. So the whole big Rube Goldberg apparatus in the middle would be transduction. Okay, good. So let's move back to the beginning of the process.
I'm sorry, your mic is breaking up again.
I was saying to move back to the beginning of the process. Can you hear me? Back to reception?
Yeah.
Okay, so that was a great diagram. But on the test, you're also going to have to explain verbally what's happening. The diagram by itself probably won't be enough. So I don't expect you to write this all out now, but let's go through this step by step and just tell me verbally what's happening in each step. So the light hits the phytochrome. That activates the phytochrome. By the way, I saw you were using the asterisks. I think that's a good thing to use on the test. If this was the test, you'd have to make a note that asterisk means activated, so the grader knows what you mean by that. But if you tell the grader that asterisk means activated, I think that would be a good approach. So anyway, after the phytochrome is activated, just describe to me in words what's happening then. Interpret to me what your picture is showing.
After the phytochrome is activated, so I'm going to scroll. When the phytochrome is activated, the G protein and GDP are going to attach to the phytochrome. Once that happens, GTP is going to knock off GDP.
Good.
And then the G protein and the GTP are going to detach from the phytochrome.
Good.
Once that happens, the G protein and GTP are going to attach to a guanylyl cyclase.
So you might want to put an asterisk on the G protein now in front of the arrow, since it's now been activated by the GTP. To the left, right? Yeah. You have, that's right, and maybe the next one down below. The GTP has activated the G protein to be consistent with the other notes. Okay, so that's good. All right, so let's see. So say again, what does the G protein do now that it's activated?
The G protein is going to attach to a guanyl cyclase.
Excellent.
Once it does that, a GTP is going to attach onto it, but then remove itself in order to create cGMP.
Okay, that's good. Now, I think we could describe that more clearly. Basically, what's happening here is the activated guanyl cyclase will convert GTP into cyclic GMP.
Mm-hmm.
That's the key thing to point out. The guanyl cyclase is changing GTP into cyclic GMP. That's what we're trying to show here in our picture. The guanyl cyclase is changing GTP into cyclic GMP.
Okay.
Good. By the way, just to help you see what's happening there, the T in GTP stands for triphosphate.
Mm-hmm.
And the M stands for monophosphate. And
Okay.
remember, what does the C stand for in cyclic GMP? Oh, excuse me.
Cyclic.
What does the C stand for in cGMP?
Cyclic.
It does. So what is the guanyl cyclase actually doing? The guanyl cyclase is chopping off two phosphate groups and then changing the molecule into a ring. That's what it actually does. That's what this is saying. GTP is losing, the guanyl cyclase is chopping off two phosphate groups to make it into a monophosphase and then forming it into a ring to make it cyclic. So it's changing the shape of the GTP by chopping off two phosphates and making it cyclic, just to help you remember. I'm not saying you don't actually have to describe that in your answer. That's more detail, I think, than they would expect. But if you know that, that might help make it easier to understand what's going on here. Good. What type of molecule is guanyl cyclase? What type of molecule is guanyl cyclase?
Enzyme?
That's right. How can you tell?
The ACE.
That's right. And it's the one that's doing this. By the way, do you remember what does the G stand for in GMP?
Guanyl?
Yeah, or guanine. And then what does the C stand for?
Cyclic.
So notice that the name guanyl cyclase makes sense. What does guanyl cyclase do? It takes this molecule with guanine and makes it cyclic. That's part of what it does. So it's a logical name. Good. Now, one thing I think you probably want to mention for full credit in an essay answer is that the cyclic GMP is a second messenger. I think that's probably a term they want you to mention.
Yeah.
So you can make a note down there that the cGMP is a second messenger. Okay. Also, something that's probably worth mentioning is that the guanyl cyclase will create, each guanyl cyclase will probably create hundreds of cGMPs. So we only showed it making one because we don't have the energy to draw hundreds of those. But after it makes one GTP into a cyclic GMP, it'll turn another GTP into a cyclic GMP, and then it'll turn another GTP into a cyclic GMP. It might do that hundreds of times. So each guanyl cyclase can create hundreds of second messengers. That would probably be something that's worth mentioning in your essay answer. Okay, so then what does the cyclic GMP do?
Oh, wow. I lost where I was. And there I am. Okay. Second, let me just scroll there. Okay, once the cyclic GMP is attached to the G protein, the guanyl cyclase, the cGMP is going to detach, and then that's when the cGMP is going to attach to a protein kinase. So we'll say A.
Good.
And that'll activate the protein kinase A, and then that protein kinase A with the attached cGMP is going to bring a phosphate over to protein kinase B, activating that protein kinase. Then that protein kinase B will attach a phosphate to C, activating C, and then however many times it has to do it, once it gets to the last one.
Excellent.
Protein kinase B with a phosphate is going to enter the nucleus.
Excellent. Very good. Let me interrupt you for a moment. So I just wanted to point out, again, in your essay answer, it would probably be worth mentioning that protein kinase A will probably phosphorylate maybe hundreds of kinase B. And then each kinase B might phosphorylate hundreds of kinase C. And then each kinase C might phosphorylate hundreds of D. So it's like there's a large amplification of the signal. Each kinase can phosphorylate hundreds, perhaps, of other kinases so that the signal gets highly amplified. And also, it would be good to mention, as you went back and wrote, that this is called the protein kinase cascade. Hopefully it's clear why this is considered a cascade. It's like one domino that knocks down 2 dominoes, that knock down 4 dominoes, that knock down 8 dominoes. It's like a big pyramid of dominoes falling down. Okay, very good. So now let's continue with our discussion. What happens next? By the way, just for your notes, it looks like the kinase inside the nucleus should be labeled protein kinase D, just to be consistent with your notes, not B. Just to be consistent with your notes. That's just a made-up letter anyway, but we just want to be consistent with what we wrote up earlier. If this was a test question, you might want to put the letters in quotation marks, just to show that they're not real names. We're just using those to help us give our discussion. Okay, good. Then what?
So the protein kinase will send over a phosphate to the transduction factor, and once the phosphate's attached, oh, sorry, I was reading my terrible handwriting. Once the phosphate's attached, it's going to activate the transcription factor.
Great.
That will then attach to a strand of DNA.
Good. To be more specific, it will attach to a gene on the DNA strand.
Mm-hmm.
Good.
And once the DNA is transcribed.
Once that, and again, it would be good to be specific. That will cause that gene to be transcribed.
Mm-hmm.
Good. Because we don't want to transcribe the entire strand that has thousands of different genes on it.
Oh.
So the whole purpose of the transcription factor is to say transcribe this one gene from the strand. The strand would likely have hundreds or thousands, I don't know how many genes on it, but the transcription factor makes sure that we're going to transcribe just one particular gene on the strand. Good.
Once that DNA gene is transcribed, mRNA exits the nucleus and enters the cytoplasm. Once that happens, the greening process begins.
Good. Let's take that one step further. What happens to the mRNA in the cytoplasm? There was one more translation. There was one more step that was listed on the slide. So what's the next step that after the mRNA is brought into the cytoplasm?
Oh,
what's the name of the process that the mRNA undergoes in the cytoplasm? This is actually pretty important.
Yeah. In the cytoplasm, the greening. No.
So what I was going for here, I actually kind of let the cat out of the bag a second ago. The mRNA is translated in the cytoplasm.
Oh, yes.
And that would be an important thing to mention. So maybe, so let's erase the line that says greening process, because we're not quite ready for that yet. The next step is mRNA is translated. That's something you studied in the first semester, that the process of genetics is first transcription and then translation. And then what's produced when the mRNA is translated? What type of molecule does that produce?
An enzyme?
Probably it would produce enzymes, but the way the book describes it is just proteins. Actually, it's likely that maybe most or all of them would be enzymes, but what we know for sure is they're proteins. So let's write down that the mRNA is translated, and that produces what the book calls the greening response proteins. So one more arrow, that produces the greening response proteins. Let's spell that correctly. That's greening.
Oh, whoops.
Okay, excellent. Let's scroll up a little bit to give ourselves a little bit more room. All right, there was a couple key things we left out, but overall that was an excellent answer. I'm sorry, let's scroll. I guess I meant scroll down. Back to the greening response proteins.
Oh, 1 second, sorry.
Good. Now, what will the greening response proteins do? Well, remember that while the potato was underneath the ground, the stem was growing very rapidly to try to reach the surface. Now that the mission is accomplished and it has reached the surface, the stem elongation is going to slow. So one thing that happens now is that the stem elongation slows. You might want to write that down for your notes. So the proteins will cause the stem elongation to slow.
Just add it onto here? Or
Yeah, you
put
might say
drawn
the
arrow?
proteins cause stem elongation to slow. Good. Remember that also that while the potato was underground, there was no point producing leaves. Now that it knows that it's perceiving sunlight, now it's going to expand its leaves. So another thing that will happen now is that the proteins cause the leaves to expand. So we can say, comma, leaves expand. Remember that while the potato was underneath the ground, it didn't need much root growth because it wasn't really ready to mature yet. Now that it's ready to actually mature, it's going to need nutrients. So now the roots are going to elongate. Notice that the stem is not elongating as fast, and now the roots will elongate more. So we can say, comma, the roots elongate. And remember that while it was underground, the potato wasn't producing chlorophyll. So now that it's above ground, it can start producing chlorophyll to absorb sunlight. So chlorophyll will be produced. So do you see why this is called the greening response? Because what color is chlorophyll?
Green.
That's right. While the potato is underground, I think it would be kind of a pale white. But once it reaches above ground, it's going to start to produce these green leaves. That's why this is called the greening. That's why this is called the greening response. You probably don't have to mention all of those on the test, but it might be good to mention one or the two of the things that the proteins actually do in your answer. Good. Okay, so overall, that's a very good approach. Let's go back to my screen.
Okay.
So that's obviously you did some good work on that. That's a very complicated process. You did some good work on memorizing that process, and you know that's going to be on the test, so that should pay off for you. However, that would be too much to write during the test, right? You might not have time to write that much. You probably won't have enough space on the page. So basically what you did there is you copied kind of what I wrote in my notes in our last session. That's great for learning, but now what we should actually do on the test is probably basically do what's on the slides here.
Yeah.
So so before tomorrow, you should probably practice boiling this down just to what's on this slide, because this is enough. This is something you could actually fit on the page. Why did I write out all the extra steps then? The reason I was writing it out in more detail was to help us understand what this means so that you can describe it in words. Because, for example, at first it wasn't really clear what was going on here. So it's good to have in your notes everything written out in more steps. For example, you don't need to write out protein kinase A, you don't need to write out on paper A putting the phosphate on B, B putting the phosphate on C. You can just write protein kinase cascade. But then in words, you should say, in the protein kinase cascade, one kinase will phosphorylate another kinase, which will phosphorylate another kinase, which will phosphorylate another kinase. You can describe that in words rather than writing it out. It would just take too long and take too much space to write it all out. So my suggestion is on the test, basically have this picture memorized and just write out this picture. And then in words, you should describe what's happening in the key steps. For example, what's happening here?
The GTP is... Oh, wow. The GTP is going to be changed into the cGMP.
By who?
The guanylyl cyclase.
Yeah. So what you should actually be, you should be drawing this picture and say, this means the guanylyl cyclase changes GTP into GMP. Good. And what's happening in the kinase cascade?
I'm sorry.
What happens during the kinase cascade?
It just cut off that cascade. Sorry. During that time, the cGMP is going to attach itself to one of the protein kinase A.
Right.
or whatever,
Right.
I'll call it A on the
Sure.
test, and that will then transfer over a phosphate to protein kinase B, and
Hundreds of
then
those. Good.
hundreds of those, and then basically the cascade will happen,
Okay,
and
excellent.
they'll
Good.
keep sending down phosphates
Great.
to other protein kinases.
That's great. You don't have to actually draw it out diagrammatically,
Yeah.
because that would just take up too much space and too much time. And then also, obviously, here's what you can draw then for the response. Basically, you should just memorize this and write this down and then describe it in words. Notice that it's important to include the translation step. That's an important step. I think that's important for you to include in your answer as well. So I'm not sure exactly how much detail your instructor expects on this, but obviously you don't want to spend the whole test on this one problem. So you have to give a reasonable amount of detail, but not overdo it so much that you run out of space or that you run out of time and you can't answer the other questions well. Okay, now remember, there's a whole other there's a whole other pathway that involves the calcium channels. We didn't talk about that yesterday, but have you had a chance to try to memorize that or learn that pathway on your own?
No, I haven't tried. I just been reviewing this.
That's fine. All right, that's perfectly okay because we hadn't talked about that. But probably we should talk about it now. So let's go in to talking about that process now, because that'll be part of the test. So let's take a look at what's happening here in this step. So in the first step, the light hits the phytochrome. And then try to describe then what do you think is happening then with this arrow in words. What do you think this arrow represents? What happens after the light hits the phytochrome? That will activate the phytochrome, so we could put an asterisk next to it. And then what does this arrow represent happening?
The inactive G protein attaching to the phytochrome.
Excellent.
And and then what's happening now is the GTP is going to kick off the GDP.
Excellent. Great. Excellent. So there's a couple different things happening. One thing that happens is that the G protein attaches to the phytochrome, and then that allows the G protein to lose a GDP and replace it with GTP. Excellent. Okay. And then what's happening here?
Okay, so the activated G protein is going to attach to the calcium. Is that calcium?
Yes, calcium.
Okay.
That's one of the elements in the periodic table. Yeah. So this is the mineral calcium. Good.
Mm-hmm. So the calcium 2 channel, so that'll open, and that'll allow the calcium molecules to enter.
Enter what?
Enter the cytoplasm.
That's right. Enter the cell. Good. By the way, one thing that's worth mentioning is what's the purpose of replacing the GDP with GTP? The purpose is that activates the G protein.
Yeah.
G proteins with GDP are inactive. G proteins with GTP are activated. So it might be a good idea when your answer to put an asterisk at this point to show that this is activated, or you can just write active. That's what allows it. So notice, could this G protein over here open a calcium channel?
No.
No, it's only when it has the GTP that has the right shape to open the calcium channel. Excellent. So then what do you think this part of the picture shows happening? It's a little bit cryptic, but take a guess. What do you think is happening here?
I think the calcium molecules are attaching to the calmodulin.
Yes.
Calmodulin.
So at first, the calmodulins are just floating around, and then they attach to the calcium. By the way, we should call these calcium ions rather than molecules because they have a charge.
Yeah.
So the calcium ions are attaching to the calmodulin. Good. So the calcium ions are attaching to the calmodulin. Good. And then what do you think this shows happening?
Well, specific kinase. Okay. They're going to attach to a protein kinase.
Good. But remember, we want to try to avoid using the words it or they. Who is attaching to the kinase?
Oh, the calcium ion calmodulin is going to attach to the protein kinase and activate it.
Good. So, by the way, what was the purpose of attaching the calcium to the calmodulin? That was to activate the calmodulin. So now we can put a star on the calmodulin. So why do they call this calcium 2 plus dash calmodulin? All they mean is that it's a calmodulin molecule that's attached to some calcium ions.
Okay.
Good. So we could say then the calmodulin is going to attach to a kinase. Good. All right. Very good. What does this arrow show happening?
The specific, no, the comodulin attached to the protein kinase is going to become, is that going to be a transcription factor?
So what is this?
That is the specific kinase activated.
Actually, that's incorrect.
Oh.
These red little globs are transcription factors.
Okay.
This is the kinase.
All right. Okay.
So this kind of a semicircle, this semicircle. Yeah. So what does this arrow? What do these arrows represent happening now? What happens after the kinase? So who is going to move into the nucleus?
The kinase.
The activated kinase. Yes.
Yes.
And then what will the activated kinase do when it's in the nucleus?
Go through the process of transcription.
Let's be more specific. Exactly how? What will the kinase do? What is a kinase? What type of molecule is a kinase?
It's an enzyme.
That's right. As you can tell from the ACE. What do kinases do in particular?
They attach a phosphate group to another enzyme.
What is this kinase going to attach a phosphate group to?
The DNA gene.
That's actually not quite right. It's going to attach a phosphate group to this little blob. And what is this blob?
That is the kinase.
Actually, no, this was the kinase. This semicircle is the kinase. The red blobs are the transcription factors.
What did we say that small blob was? The calcium ion?
Let's see, I missed you. So this is a transcription factor.
Yeah.
This is a transcription factor that has a phosphate attached.
Yes.
This is another transcription factor, and this is the same transcription factor after the phosphate has been attached. These little circles are calcium ions.
Yeah.
These little circles are calcium ions. And what's this dumbbell-shaped thing?
The calmodulin.
Yeah, this is the calmodulin. And what's the semicircle-shaped thing?
The kinase.
That's right. So that's what all these shapes are representing. Does that make sense? Do you have a question about that?
Okay. So the kinase is going to enter the nucleus.
Right.
And then is it going to give a phosphate to the transcription factor?
That's right. Can you see that happening in the picture here? Because what does this red glob represent?
Transcription factor.
That's right. Good. Can you see this? Now here we have the same blob, but now it has a P attached. So the picture is trying to show you that the kinase has attached a phosphate group to the transcription factor. So let's review that again. The kinase, so who is moving into the nucleus?
The kinase.
Yeah, the kinase moves into the nucleus. What will the kinase do once it's in the nucleus?
It's going to give a phosphate to the transcription factor.
Excellent. Yes, it will give a phosphate to the transcription factor. Then, what will the transcription factor do once it's been activated by the phosphate?
It's going to transcribe the DNA gene.
That's right. It will attach to a specific gene, and that will cause that to be transcribed. Do you notice how some of these pictures have, can you see that some of these pictures have a yellow border around the shape?
Yes.
Yeah, the yellow border means activated. For example, notice here we have a G protein with no yellow border. But then after the GDP turns into GTP, they added the yellow border to show that's been activated. Or notice here, this kinase has a yellow border to show it's been activated. Or notice here, what does this blob represent?
The transcription factor.
But notice now, there's now a yellow border to show that it's been activated. So we said that the transcription factor is going to attach to a gene. That causes the gene to be transcribed. Good. By the way, do you remember what's the product of transcription? When a gene is transcribed, that produces mRNA. Good. And then what's going to happen to the mRNA?
The mRNA is going to exit the nucleus and enter the cytoplasm.
Excellent. That's what this arrow is representing. Great. And then what process does the mRNA go through?
Translation.
And that produces...
I'm trying. What is it?
You can just follow along here.
Oh, the greening process. I thought
the greening response proteins. Good. And then you might want to add, those do things like they cause the stem to stop elongating so fast. They make the leaves expand. They produce chlorophyll. They make the roots elongate. You might add a little bit about that. Okay. So hopefully now that's our second pathway, and we understand that a little bit more clearly. And again, for the test, the best thing is just to memorize this picture and then describe in words what's happening. So let's go through that pathway again. So what was the first step of this pathway? What was happening here? When you're ready.
Okay, so the light hits the phytochrome.
Good. Then what?
Oh, are we going from
Let's
the
do
first?
the other. I think you understand the upper pathway. So let's go
Okay.
through the lower pathway again.
Okay, so once the light hits the phytochrome, the inactive G protein is going to attach to that phytochrome. And once that happens, a GTP is going to knock off the GDP, activating the phytochrome.
Activating who?
The phytochrome?
Actually, the phytochrome was
the
activated
phytochrome
here.
activates the protein.
Activate. Say that one more time.
The phytochrome will activate the G protein?
Yeah. That's what they show here. Good.
Mm-hmm. Okay, so once the G protein is activated, it's going to detach itself from the phytochrome and move over to the calcium ion channel. And that allows the calcium ion, because of the activated G protein, it'll allow the calcium ion channel to open,
Great.
letting in calcium ion molecules. These calcium ion molecules are going to attach themselves to the calmodulin
Molecule. Good.
molecule, and then that calmodulin molecule will attach itself to the kinase protein, activating it.
Excellent.
When the protein kinase is activated, it's going to enter into the nucleus and give a phosphate over to the transcription factor, then which it's going to attach itself to a DNA gene and go through the process of...
Who's going to attach to the gene?
Oh, it's going to... the transcription factor will attach itself
Excellent.
to the DNA gene.
Good.
And go through the process of transcription, producing mRNA. The mRNA will then leave the nucleus to go through the process of translation, and after that, the mRNA will go through the greening response proteins. It'll create greening response proteins.
Let's clarify that last step. So what's the input to translation? The input is mRNA. And what's the output from translation?
Greening response proteins.
That's right. That was a little bit unclear. So the mRNA will be translated, producing greening response proteins. And then those would cause things like chlorophyll to be produced. Okay, very good. So, good. So what are these little circles? What do they represent?
Calcium ion molecules.
Yeah, and what's the dumbbell?
Modulin.
That's calmodulin. Good. And what's the semicircle? So I was asking you, what does the dumbbell represent, if you can see my screen?
The calmodulin.
Oh, yeah, the dumbbell is the calmodulin. And then I was asking you, what does the semicircle represent?
The transcription factor. Oh, I'm sorry, the protein kinase.
Good. And what do the blobs represent?
The transcription factor.
Okay, good. So those are all things that's important to be familiar with. So I think the best thing is just memorize this and then copy it exactly on the test, and then add some words to explain what's happening in the steps. That should be a good answer for this type of problem. So remember, again, the diagram that you made for this step would be way too long for the test. You need to boil it down to these steps here, or otherwise you'd run out of space. All right. So we know this is going to be on the test, so why don't you take 30 seconds or a minute and try to memorize this lower pathway. Take a minute or two and try to memorize this lower pathway, because you know you're going to have to talk about it on the test. So take your time, but tell me when you're ready to keep going.
All right. One second.
Take your time.
Okay.
Okay. Well, let's move back to your screen then. Turn on your screen sharing. Oh,
Okay.
good. I just wanted to point out one thing. Did you notice one difference between these two pathways? In the upper pathway, we have a kinase cascade. One kinase activates another kinase, which activates another kinase, which activates another kinase, which activates another kinase. Notice there's a difference. In the lower pathway, we apparently only have one kinase. So that's one distinction that's worth noticing. Here, there's just one specific kinase, not a whole cascade. Okay, so let's try turning on your screen sharing. And now, so now let's try, if the problem was to describe the signal transduction process for the greening response, let's try writing down the lower pathway now.
All right. Okay, so the light is going to, well, this is the cytoplasm. The light is going to hit. I'm just remembering. So the light will hit the phytochrome.
Excellent, good.
And then I'm going to have our okay, and then so next is the Phytochrome, but then GDP is going to leave and GTP will enter. This is going to cause to be activated.
Good.
Okay, so okay. Oh well, I'm trying. Okay, so each protein will attach, open up the calcium ion channel. These are calcium ion. Okay, and then they
Good.
will.
You might want to show maybe one calcium actually moving through the channel. Maybe put a dot outside the channel and then an arrow to show it moving through the channel. Maybe an arrow going through to show that they're moving through. All right, excellent.
And then
where is the calcium channel? What part of the cell is the calcium channel in?
it's would it be? Is it like it's inside? It's on the cell wall, I guess.
Good. Yeah, it would be in the cell membrane.
Yeah.
Because remember, it's supposed to allow calcium to enter into the cell. So the calcium channel is located inside the plasma membrane. Good.
Okay.
You might want to draw a line then to show that it's inside the plasma membrane, just like a horizontal line. Yeah, good, exactly. And you could label that as the plasma membrane. So it basically crosses the plasma membrane. It's basically like a tunnel that lets the calcium cross the membrane. The calcium cannot cross the membrane directly, so they need a kind of a tunnel to get through.
Okay.
And you've shown that now it's open. Good.
Yes.
Before the G protein attached, the two parts would have been closed, so the calcium couldn't get through. Good.
Okay, so their calcium is going to attach to the calmodulin, and they're going to no. This is
Good. What does calcium 2 plus calmodulin mean? It just means a molecule of calmodulin that has a bunch of calcium stuck to it. Good.
Alright. So once that happens, so the calmodulin is going to attach to and that's going to activate the protein kinase. Okay. Guess I'll just show. It's going to enter the nucleus. This is the transcription factor. Could I put TF as short, or should I write it out so I remember it?
That would be fine. So you could label that as TF. That's a great idea. If this was a test question, you could just say in your answer, the protein kinase is activating the transcription factor, and then obviously the reader would know that you know what that is. Good.
Mm-hmm.
But yeah, it's obviously a good idea to use some useful abbreviations to save some space and some time.
And so the protein kinase is going to give a phosphate to the transcription factor. So
good.
And then I'll start transcription.
Great. What has the transcription factor attached to?
The DNA gene.
Good.
So once transcription happens, mRNA is produced.
Excellent.
And then the mRNA will exit the nucleus. And then translation. After translation, screening, create screening response. Proteins. Should I list one of the the green response proteins can make chlorophyll? Or
that would be, yeah. One thing they do is they make chlorophyll. Anything else they do? Or they cause chlorophyll to be produced. Anything else they do?
Oh, wow.
One thing I would especially focus on is they cause the leaves to expand. Remember, that's why this is called greening.
Mm-hmm.
They're called greening because they cause green leaves to be produced. Green leaves with chlorophyll. I think that would be fine for the test. That's enough. That helps us to see why it's called greening. Why are the leaves green? Because they have chlorophyll. So those two things go together. And why is now a good time to produce green leaves? Because the light... The plant has sensed light. Remember, the whole starting here is that the plant sensed light. There's no point making leaves until you sense light, because the whole point of the leaves is to absorb light for photosynthesis. That was something you probably studied in the first semester. Okay, excellent. So I think that's some good progress on that. I think we have that down for right now. I'm going to go back to my screen.
Okay.
So obviously you want to practice that a couple more times before the test, since you know that's going to be on the exam. It would be easy to start forgetting it, but for right now, I think that would be good for a full credit answer. I just wanted to point out here, notice here in the picture, here they show the calcium channel in the plasma membrane. So just to give you a picture of what's going on here, in this picture it's closed, but then after the G protein hits it, it opens up. Okay, just to give you a better mental picture, but you did a good job of diagramming that out. Okay, so since you know some of the things that'll be on the test, you can just spend some time today really making sure that you have those answers down. Like I say, I'm not 100% sure. I'm not 100% sure how much detail your instructor is expecting. But if you have this memorized and you can explain it, that should be enough for full credit, or more than enough, basically, because there's not that much space. I just wanted to. So how would this be tested? So we just suppose there was a question that said, describe the signal transduction in plants in regards to the role of phytochromes. Describe the signal transduction in plants in regards to the role of phytochromes. Well, that's what we were just talking about, right? Mm-hmm. So that's when you would go through the two pathways we were just talking about. You would talk about the G proteins and the protein cascades and the calcium and the modulant. So what we were just going through was the signal transduction in plants for the role of phytochromes. That was what we were just covering. That was one of the learning objectives from that chapter. Or they might say, describe signal transduction for the greening response. Well, that would be the same exact thing, right? If they say signal transduction for the greening response, again, it would be the same thing. I just wanted to mention one other name for this. This is called the greening response. It's also called de-etiolation, just in case they use that term on the test. Mm-hmm. So if they said, describe signal transduction for de-etiolation, it would be the same thing we were just talking about. Signal transduction for de-etiolation is that same multi-step process with the phytochromes and the G proteins and the protein kinases. These are just different ways they could phrase the question. These videos are offered on a pay-what-you-like basis. You can pay for the use of the videos at my website. There's a link to my website in the annotation. The address is www.freelance-teacher.com/videos.htm. Or you can just use the link in the annotation. Thank you.